Okay, hello everyone, and welcome to this session on NF2 and Schwannomatosis, bench to bedside. We've got 4 talks for you, and I'm going to start off with the first one by introducing Anna Sablina, who is from Leuven in Belgium, and she's going to be talking to us about her work on LZTR1 in Schwannomatosis. So over to you, Anna.
Okay, first of all, I would like to thank organizers for inviting me and giving me an opportunity to share our latest results on LZTR1. LZTR1 was first identified associated with schwannomatosis in 2014 by a group of Ludwig Messing. And a few years later, germline mutation of LZTR1 were found to be associated with Noonan syndrome, which is a rosopathy disorder, also associated with increased activity of the RAS signaling pathway. So what is the current status of LZTR1 mutation in schwannomatosis patients? About 30% of these mutations are missense. 20% of these mutations affecting splicing of LZTR1 and completely destroy production of functional LZTR1 protein. And about 40% of these mutations are nonsense or frameshift. So taken together, they suggest that the majority of schwannoma-associated LZTR1 mutations more or less completely disrupt expression of LZTR1. On the other hand, we and others found that LZTR1 work as a component of the LZTR1 calin-3 complex that can ubiquitinate and inhibit the activity of the RAS signaling pathway. So, and then we kind of decided to see actually how all this, we can combine all this together and see actually how loss of LZTR1 function could contribute to schwannoma development. And as a first model, we were using primary human Schwann cells. And what we found that suppression of LZTR1, which is mimicking loss of LZTR1 activity in schwannomatosis patients leads to gene expression signatures that resemble gene expression signatures of proliferating Schwann cells during nerve regeneration. So it means that loss of LZTR1 induces dedifferentiation of Schwann cells. We also find that LZTR1 loss can promote the growth in 2D and 3D of primary Schwann cells. And consistently to our observations that the LZTR1 calin-3 complex can ubiquitinate RAS proteins, we found that loss of LZTR1 in Schwann cells leads to increased levels of RAS ubiquitination that we detected by proximity ligation assay using anti-ubiquitin and anti-pan-RAS antibodies. And consistently with decreased levels of RAS ubiquitination, we could see an increased level of RAS activity in Schwann cells with LZTR1 indels. And we also observe, in addition to increased RAS activity, we also observe increased activity of the MAP kinase signaling. As you can see, in serum stimulated Schwann cells harboring indels of LZTR1, we could observe an increase in phospho-MEC and phospho-ERG. By the way, in this assay, we did not observe an increase of phospho-AKT. So altogether, this suggests that loss of LZTR1 expression can promote dedifferentiation of Schwann cells and can trigger their growth due to dysregulation of RAS ubiquitination and increase the RAS signaling pathway. So, but what is about missense mutations? We are also look how missense mutation could contribute to schwannomatosis development. As you can see here, the identified mutation associated with schwannoma spread out through the whole LZTR1 gene. But as I mentioned already, LZTR1 serves as a part of the big ubiquitin ligase complex. So each part of the protein actually is essential to interact with the different components of this ubiquitin ligase complexes. So we started step by step to see how specific mutation in the specific domain could affect these interactions. So we started with the Kelch domain of LZTR1, which is responsible for substrate binding. And in our case, we believe that LZTR1 controls ubiquitination of RAS proteins. So what we found when we look for specifically for the Kelch domain schwannoma-associated LZTR1 mutant, what we found that when we have wild-type LZTR1, we have a quite nice interaction between LZTR1 and pan-RAS. While all disease-associated mutations of LZTR1 located within Kelch domain dramatically decreased these interactions. So then we were looking to the BTB back domain of LZTR1, which is responsible for dimerization and binding to calin-3. And again, when we look for two mutants, which is recurrent mutants both in schwannomatosis and Noonan syndrome, we could see that this mutation dramatically decreased the ability of LZTR1 to interact with calin-3. And we also find one mutant, 812, which did not affect the interaction of LZTR1 with calin-3, but affect LZTR1 dimerization. While this dimerization is really essential to form a fully functional LZTR1 complex. And what is also amazing that all BTB back domains, BTB back domain mutants also dramatically affect subcellular localization of LZTR1. As you can see here, wild type as well as all Kelch domain mutants form so like puncture. But all tested BTB back domain mutants showed diffuse cytoplasmic localization. So taken together, they suggest that all these disease-associated LZTR1 mutants are loss of function. So we were able functionally confirm this again by doing rescue experiments. As you can see here, when we analyze the anchorage-independent growth of schwannoma cells with LZTR1 indels, when we overexpress wild-type LZTR1, we could see a nice decrease in the ability of the cells to grow in soft agar. While we did not observe such effect when we analyze cells with overexpression of different LZTR1 mutants. And we also observe increased phosphorylation of ERK in different schwannomas harboring LZTR1 mutations. So this data, mostly of course in vitro, suggests that in fact LZTR1 could contribute to schwannomatosis development. But of course, we would like to have a proof whether it could be also be modeled and could be seen in vivo. So, and from this, our in vivo story started. So we generated LZTR1 knockout mice. It was just a full knockout. Unfortunately, LZTR1 was sufficient to partially recapitulate many of Noonan syndrome phenotypes. As you can see, this mice is smaller. It has a shorter snout. And it also showed quite dramatic cardiac hypertrophy phenotypes. As we can see at histology level, it's mostly due to increased size of cardiomyocytes and also due to, by echography analysis. And it's quite logical because, as I mentioned before, germline mutation of mutations of LZTR1s are also associated with Noonan syndrome. Of course, we were expecting to see some cancer-related phenotypes in our mice, but even with monitoring these mice quite carefully during the whole their lifespan and did multiple tests, we couldn't detect any cancer-associated phenotypes in this mice. So that's why we started to see how actually we can model LZTR1 associated schwannoma. And for this, I'm going back to human, the data from human genetic, and just to say that LZTR1 mutation is not only one observed in LZTR1 associated schwannoma. So basically, the first event, it's believed that it could be a germline mutation of LZTR1. But then in all schwannomas, this germline mutation is associated with loss of 20q. But also quite commonly, there is a second mutation in NF2 that is also found only in schwannomas, but not in blood of this patient. So we decided to try to model this situation. And as a first model, we use tamoxifen-inducible nestin-3 system. Nestin is expressing quite early on during development in neural crest cells. And we generated mice in which one LZTR1 allele was completely deleted, kind of to mimic germline mutation of LZTR1, and the second allele was fluxed. So, but when we use nestin-3 and induce it during pregnancy, we couldn't get any pups. So it looks that this was also embryonically lethal. We also tried to did the induction at weaning 3 weeks after birth. And to our surprise, we still were able to see that in this case, we could see some LZTR1 knockout in our cells. Here we characterize CD45 negative O4 positive cells. And again, we started to monitor these mice for about 2 years, but couldn't detect any schwannomas. So we also tried to induce schwannomas using irradiation because it's quite well described that schwannoma tissues are commonly occur in adults who went through radiotherapy in their childhood. So we performed several focal irradiation of mouse heart. It was 5 times over 3 grays. And again, we were monitoring these mice. Unfortunately, again, even we were monitoring for 40 mice, no tumors were detected. So, and then we decided to a more specific Schwann cell model. We choose and decided to go for P0 Cree. In this case, we generated P0 Cree and got a complete loss of LZTR1. Again, we were monitoring this mice and got no schwannomas. So, but as I told that LZTR1 mutations are always accompanied by 20q loss, and in some cases also mutation of NF2. NF2 is a coding gene, coding protein called Merlin, which is a quite well described components of the KIPA pathway. So that's why we decided to see whether LZTR1 loss could cooperate with KIPA signaling to induce schwannoma development. So just for us, it was, instead of using NF2, was much more handy to use LATS1, LATS2 knockout mice. So we generated P0 Cree with LZTR1 flox and a loss of one allele of LATS2 and one allele of LATS1 and one allele of LATS2. So, and even after the growth, we could already detect that there were some pups with peripheral nerve tumors. So, suggesting that in fact LZTR1 could cooperate with LATS1 and 2 to induce schwannoma development. We couldn't detect such tumors in either LZTR1 knockout mice or in LATS1, 2 in mice where both LATS1 and 2 were deleted. But of course, it was just a few mice showing such severe phenotype. So penetrance for this is, I would say, quite low. So we started to monitor our other mice. And we were quite happy to see that at the age around 6-7 months, when we did MRI, we could see a quite significant enlargement of vestibular cochlear nerve. And when we again did the histology of this mice, again, we could see that this nerve is completely enlarged. So unfortunately, I'm not a real expert in the field, and actually I would like maybe to use an opportunity here. And if somebody interested to help us and collaborate with us, to help us analyze these MRI images and to help us with histology analysis or any advice, I will really appreciate, because it could really help us to move forward with the analysis of these tumors. So please, I will really appreciate if you contact me. So I would say any help or any advice, I will be really grateful to get. So for now, what we are planning to do, we are planning to isolate Schwann cells from these mice by purifying CD45 negative or 4 positive cells, and to perform integrative transcriptomic and proteomic analysis to figure out how actually LZTR1 loss could cooperate with the HIPA pathway to induce schwannoma development. So, and to finish this, yeah, I'm describe our efforts to model LZTR1 associated schwannoma. I feel like we are moving forward now. especially I hope to get some feedback and help of the community. And I would like to thank you all my collaborators and my funding body. And I will be happy to get your question.
Okay, that's great. Thanks very much. So we have time for maybe one or two questions. There's already a question in the Slido, so from Gareth Evans. How do you explain the high rate of loss of function LZTR1 pathogenic variants in NOMAD, which is around 1 in 410, but a low incidence of LZTR1 schwannomatosis in the general population, so less than 1% of germline loss of function variants cause schwannomatosis?
Yeah, it's a good question. So because at the beginning, when there were kind of all mutations were not so well characterized, so the idea was that maybe different mutations somehow triggering either Noonan or Schwannomatosis. But now it looks like there are plenty of overlapping mutations. So genetically, it's completely unclear why there is such what is actually going to which specific mutation will lead. So I have, unfortunately for now, I do not really have explanation for this question.
And there's a question in, is there a genotype-phenotype correlation that would predict Noonan syndrome versus schwannomatosis phenotype associated with LZTR1 pathogenic variants?
So still kind of, yeah, it's more or less related to the first question. In Noonan syndrome, so it could be kind of two scenarios, or it could be just a deletion in one or mutation in one allele of LZTR1. So usually it's a mutation in the Kelch domain, and people believe that in addition to loss of function, as I explained, it's a loss of ability to interact with RAS. It could be some additional dominant negative effects, but nobody really explore this possibility. The problem is that LZTR1 is quite difficult protein to work with. Even I have quite a lot of experience working with a lot of proteins. This one is really tough one somehow. Like purification, all biochemical studies are really problematic. Yes, that's kind of, and there are some differences in this mutation. BTB back domains in Noonan syndrome also quite commonly associated with the loss of the second allele, usually also due to the loss of some pieces of chromosome 22.
Okay. And so would you then not expect that schwannomatosis type surveillance would be suitable for people with Noonan syndrome? I guess it's more of a clinical question.
So gliomas were described, but schwannomatosis or schwannomas, no, not really.
Right. Yeah.
No, this is a mystery. Yeah.
Okay. I'm just thinking, in the interest of time, there are a couple of more questions. I wonder whether I should send them to you separately so you can answer those, and we can move on to the next talk. So then for the next talk, I'd like to introduce Barbara Rivera-Polo from Spain, and she's going to be talking to us about DGCR8, so a new gene that's been found to be associated with schwannomatosis as well. So over to you, Barbara.
Thank you. So thank you, everyone. I would like to start by obviously thanking the organizers for inviting me to my first, but surely not last NF conference. As Miriam told you, I'm going to talk about what we believe is a new syndrome that features schwannomatosis and familial multinodular goiter, and how we got into this, or how I got into this. I tend to like to start this talk with this picture, because usually one finds easier things that it's looking for and not things that it's not looking for. And I wasn't looking for the schwannomatosis gene. Same way that I wasn't, that I look at the Gioconda, I was looking for the reason of her enlarged neck, and it seems to be because she might actually have an MNG, so multinodular goiter. So with further delay, I have no conflicts. And I'm going to start by talking a little bit about Dicer. So Dicer syndrome, it is a hereditary condition that predisposes for the development of more than 15 tumor types. Some of them very, very rare. And others as common as multinodular goiter or thyroid nodules that are not actually a tumor, but an overgrowth, and affects mostly children and young adults. So Dicer is a microRNA processor. It cleaves a precursor of a microRNA into a mature microRNA that is further loaded into the Argonaut and regulate transcription there. And as I said, there was like more than 15 tumor types. Leanne DeCock is the most talented PhD student I've ever met. She worked very hard to characterize many of this. I want you to have a look at those in brown: multinodular goiter, Wilms tumor, pineoblastoma, and differentiated thyroid carcinoma. And notice that none of this is actually a schwannoma. So the first time that the folks team discovered the association between familial multinodular goiter and thyroid 1 germline mutation was in 2011. And so far, right now, that was the second or the third phenotype associated to Dicer. And this is the landscape of how mutations distribute in Dicer, right? So there's germline mutations along the entire gene, and then the hotspots are clustered here in this RNase 3B domain, so the domain that cleaves the microRNAs. Why I'm telling you all this? Well, when I started my postdoc, I was trying to look for the reason of FMNG families that were not explained by Dicer mutations, like this one. So this one was a 3-generation family affected by multinodular goiter. And then there was this woman here with 3 schwannomas and 2 variant desmoids. And I thought that this was just a red herring. And what I was interested in was the MNG. And then there was this kid here diagnosed with a cortical plexus papilloma and a disorder in the spectrum of autism. Again, my goal was to uncover the gene, the cause for this familial MNG. And my surprise is that after a few other experiments, we performed a WES, and there was one variant perfectly segregated with the disease in the 3 generations. And it was a variant in a gene called DCR8 that has never been described in the germline. This one here, glutamic 518 to lysine. And who is DCR8? Well, it is another microprocessor. It's right in the middle of the microRNA biogenesis pathway, and it helps the microprocessor, so drosha, to cleave a primary microRNA into a precursor of a microRNA that will get further exported to the cytoplasm and cleaved by Dicer. So it's also in the middle of what we call the microprocessor that today, very recently, we know that is formed by 2 molecules of ERH, another protein, DCR8 and drosha, and one of drosha. DCR8 does not cleave, but helps securing the hairpin there. So we found this alteration and we said, okay, let's see what is in the literature. We gathered all the alteration in literature and in databases in DCR8. We filtered them with our same criteria that looking for putative drivers. And we plot everything that was likely pathogenic in the germline. And as you can see at the bottom, this is a small skin of the protein. We have only the family 1, that it was our family, and then this other variant that is, I'm not going to go into this, it turned to be a polymorphism. And then in the top part, there are all somatic variants in DCR8. And there's a very clear hot spot here in the middle, clustering in the first RNA binding domain, and it corresponds to actually the same variant that we found in the germline of our family. This time, it is a somatic variant, and all these blue dots are Wilms tumors. And those red dots are actually PTCs, papillary thyroid cancers. So this is how the family ended up being after the second visit in the clinic. We interviewed them all again. This woman that I've told you before had 3 peripheral schwannomas. It so happened that has developed up to 9 peripheral schwannomas to date. We asked them, do you have any little bump or lump that is painful? And they started to say, yes, I do. Yes, I do. So after a clinical revision, 5 of them had already developed peripheral schwannomas. And then in blue, you see the bubbles with the germline variant. And then in red is all the bubbles, all the tumors that we gathered. We saw that there was presence of LOH at the tumor or at the tissue, the MNGs, except for 1. Okay, there was no evidence of vestibular schwannomas. This is just a modeling of a prediction of what the mutation would do to the binding of the RNA. This is the helix where the mutation is located. These glutamic acids stabilize this arginine and creates bonds, hydrogen bonds to the RNA. When you change by a lysine, these bonds are predicted to be broken and the arginine is destabilized. But who is also DCR8? It's not only in the middle of the microprocessor, it's also in the middle of 22q11 region, right besides LCTR1 and in the middle of what we know as the DGeorge critical region. Here, hence its name, DGeorge critical region 8. So it was also a perfect candidate for the schwannomatosis, and you know all this very well, much better than I do, and it's the 2-3 hits story in the schwannomatosis. So we had the first hit, the germline, we thought it was the first hit, the germline variant, and then we look at the tumors. This is just some of the tumors that we gathered, and we did a genome-wide analysis. As you can see, in the first line is a germline, then the second one is a tumor, blue versus red, and then for each of them, this is germline, 4 tumors of the same patient, and so on and so forth. Some of them are schwannomas, a CPP, some PTCs, and Wells tumor. And as you can see at chromosome 22, very little here, all the tumors seems to have allelic imbalance, and actually we look and there's LOH, some of them copy neutral, but most of them just a deletion of the whole chromosome. But then we thought, okay, this gene is a microRNA processor. So what's happening with microRNA profile? How can we prove that this variant caused a disturbance in the microRNA processing? So we did a nanostring profiling of different types of tumors or tissues, including orange PTCs, red MNGs, blue schwannomas, with the mutation E518K, or without. We also gather controls. And this is a hierarchical clustering performed by Javad, a very talented computational biologist in the team. And as you can see, mutant clustered together against non-mutant cases. So this variant was overreading somehow the tissue specificity of the microRNA profile. To validate this, we then went ahead, same readout, mature microRNAs, but a different sample set. We're comparing now schwannomas with a mutation in green versus Wilms tumors with a mutation, but sporadic cases in this yellowish color. And as you can see, again, mutated tumor, here in pink, they cluster together independently from the non-mutated ones. This is consensus clustering of 1,000 repetitions. And there was a total of 190 differentially expressed microRNAs in common between those Wilms tumors and schwannomas. So this mutation was actually impairing a specific profile. Right, so we have a profile, but we wanted to dig into what microRNAs are actually differentially expressed between mutated. This is schwannoma sample set between schwannomas with the mutation and without. So in red, in this volcano plot, p-value is in logarithmic scale. Fold change is here. FDR is set up at 1%. And in pink, all the dots that represent microRNAs that are overrepresented, this is relative values, in mutated DCR8 schwannomas compared to the wild types ones. And the ones that are underrepresented in blue are the ones that we are looking for, because those will be the ones that are not being processed. And I'm highlighting MIR30 here, one of the top ones in significance, for you to keep track of one of them. So microRNAs are produced in the canonical way, as I just explained you. So a primary microRNA is cleaved by the microprocessor into a precursor, further exported and cleaved by dicer into a mature duplex. But also intronic microRNAs, what are called mirtrons, are encoded in the introns of coding genes, and they are independent from the microprocessor, but dependent on dicer. They're cleaved by the spliceosome by debranching and then further cleaved by dicer. So how can we use this to further prove that our microRNA defect was due to DCR8 failure? So we look at where are the mirtrons. So when we repeat the same experiment that I just explained, the comparison between the microRNAs in mutated versus non-mutated samples, and now we look for mirtrons, they are all these black dots that are always underrepresented. I know this is very small, but I can promise you there was no significant differentially expressed mirtron that it was in the underrepresented side of the table. Why? Because they are independent from the microprocessor. This is schwannoma, but same happen when we look at the Wilms tumors. Mirtrons are all overrepresented here. And to test the pipeline, what Java did is he gathered pituitary blastomas that are tumors with dicer1 mutations and now plotted mirtrons, and they are both in the overrepresented and the underrepresented or underexpressed, we can assume, side of the volcano plot. Why? Because mirtrons are dependent from dicer. So we're still far from the defect because we're reading mature microRNAs, and what the microprocessor does is cleave into precursors. So we said, okay, let's try to change the readout and look at precursors. This is easier to say than to do. You need to sequence very deep, and also the structure is very labile, but we still went ahead and tried. As you can see, our volcano plot is very wide here. There was only little precursors of microRNAs that reached significance among those differentially expressed. Still, mirtrons black are always overrepresented, and MIR32, again, it's here. So it was the perfect candidate to go in vitro and prove what was the effect on the microprocessor. We went ahead and got a primary microRNA, incubated with a wild-type microprocessor formed by DCR8 wild-type and DROSHA wild-type over time, and we saw that we can cleave a precursor, that is this band here of 65, around 65 nucleotides. Whereas if we do the same with a defective microprocessor, with a wild-type DROSHA, but a mutant DCR8, there is no precursor. All right, so we have a defective process in the microRNAs, down, mature, and precursors, so what is going to be up? So this is very still preliminary because we only have 5 mutated samples at that time and 4 controls, but we look at targets. 2 of the most overrepresented targets were RAS proteins. Obviously, after Dr. Sablina's talk, I don't need to explain the multiple links between schwannomas and schwannomatosis genes like CTF1 and RAS pathway. And even the last paper, genomic landscape of schwannomatosis, show an LCTR1 germline schwannomas to be linked to an overexpression of MAP kinase pathway. Well, we then search for other patients. Let's see if this is an actual syndrome. And I look into schwannomas, and I couldn't find any mutant in DCR8 and thyroid nodules, and I couldn't find any. And then I said, hold on a minute, let's look for what we believe is actually the constellation of the phenotypes. So let's search for peripheral schwannomas and thyroid nodules or thyroid tumors within the same person. That was the most stringent criteria, or in on a family member. And we ran into this family. He is a young adult with 7 peripheral schwannomas, PTC and MNG, and his dad has sebaceous cyst, sister has a giant cell nerve shed tumor, and he was a carrier indeed of the same mutation. Again, we were very lucky, very surprised. This is how the pedigree looks like now. Seems to be de novo, nor the father or the sister are affected, confirming also that those are 2 features that go together, MNG and schwannomatosis. 5 tumors that we gathered from him, schwannomas, they all had LOH at the mutation spot. This is how our phenotype table is going right now, or our apple tree. MNG, schwannomatosis, PTCs are all associated with germline mutations, obviously only 2 families so far. Wilms tumors or pineoblastomas are associated with alterations DGCR8, but only somatically that we know so far. Again, only 2 families. We don't know about the George syndrome or any autism disorder. And kidney cyst, we also don't know, quite common. Not sebaceous cyst, it seems, and not giant cell shed tumor so far from the second family. But are there other phenotypes to be discovered? Well, I would like to take the opportunity, and if anyone see a patient that is reminiscent of this, please get in touch. I'd love to collaborate and get more samples so we can get to the point and learn more about this novel syndrome. I also perform a West analysis on all the schwannomas that we've gathered so far. This is just for the last 5 ones. They all seem to have mutations in NF2, somatic mutations in NF2. The LOH seems to be confirmed in NF2 too, except for this one. We plot CMVs along chromosome 22. The lesions are predicted to be copy number of 1, and when you overlap them, this green bar is DGCR8, this yellow bar is NF2, and it seems that the entire 22q is also deleted. And finally, so trying to solve how DGCR8 is going to fit into this model, DGCR8 first mutation germline is here. They all lost secondary copy of chromosome 22. And so far we had 10 schwannomas that had a secondary somatic mutation NF2, 4 that do not, about a third. And in 2 of those 4, we indeed had enough tissue to go and run a Western, and we were able to see Merlin. So actually they there is a proportion of that that seems to be okay without mutations in NF2. And I want to finish by saying thank you to all the collaborators. My very young lab, we opened the lab right before the pandemic. Always interested in postdocs that want to join the team. Clara and Sophie are working in DCRA. We're part of the hereditary cancer program and work very close to Dr. Fope's lab. And obviously all the funding bodies. And thank you all for your attention.
Okay, great. That's fantastic. Thanks very much. So I'll just have a look in the questions here. So there's one question from Oliver that says, can you detect altered microRNA profile in blood of DGCR8 mutant families?
Right. So we did the experiment. We did the experiment and we didn't seem to see any differences there. We ran it actually in a bunch of carriers, but the profiles were clustering by sex rather by their mutant versus wild type status. So it doesn't seem to be the case in the lab.
Okay. And so do you have a sense yet of the mechanism? I know you've done a little bit of looking at somatic NF2, but in terms of whether this works in the same way as LZTR1 or SMART-B1 schwannomatosis? So we do not know. We're working on the molecular. We're doing some proteomics. It's a very good question. It's very hard because DGCR8 is quite less understood than DICER is, and we have obviously all the microRNA story here that fits with DICER and DROSHA, and that can explain the MNG, but not the schwannomatosis. So I tend to think that it's going to be similar to SMART-B1 in a sense, because I tend to think that the mutation might just be not completely inactive, but selective of some microRNAs, because we can see that some microRNAs are still clean. So the lab is trying to work towards what microRNAs are actually the key ones here. Okay. That's great. There's another question has just appeared. Okay, does the type of pathogenic variant in DGCR8 is it associated with the differences among the phenotypes, genotype-phenotype associations?
Right. So in pineoblastomas, yes. So I guess the question is towards what kind of mutations are there. So that we know that is really pathogenic. The only proven one is this one. The curiosity here is that in the germline, so far, the phenotypes that we've seen is the one that I presented to you, mainly MNG-thyroid tumors and schwannomas. Somatically, it's Wilms tumors, and it is the same mutation. Pineoblastomas, though, are the only tumor type that can live or survive, let's say, with a complete loss of one of the microRNA processing members, diceryl, decyryl, or Georgia. So the genotype-phenotype correlation might be around there. Time in development where you need a particular microRNA and tissue type.
Okay, that's great, thank you. I think that's all of the questions we have so far. So thank you very much again, and I'm going to hand over to my co-chair to introduce the next speaker. So Matthias.
Well, thank you, Miriam, for kicking off the session and introducing the first two speakers. I'm now very excited to present to you two talks from members of the Fernandez Ballet Lab. And we're going to start with Maria Martinelli, and she is going to talk to us about preclinical assessment of fimipinostat, a dual phosphoinositide 3 kinase histone deacetylase inhibitor, as a therapeutic for NF2 schwannomas.
Hello, my name is Maria Martinelli. I'm a first-year graduate student in the Fernandez Valley Lab at the University of Central Florida. Thank you to the coordinators for organizing the annual NF conference and inviting me to present our data. We have no disclosures, and this presentation contains the compound currently in clinical trials for CNS tumors. NF2 is a rare genetic tumor predisposition disorder of the nervous system. NF2 patients can develop schwannomas, meningiomas, and ependymomas. Bilateral vestibular schwannomas are diagnostic for NF2. This lab focuses on development of therapeutics for schwannomas. NF2 is caused by loss of function mutations in the NF2 gene. NF2 is a tumor suppressor gene that encodes Merlin, an adapter protein that regulates multiple signaling pathways involved in cell proliferation, survival, growth, and morphology. Some of these include the RAC-CDC42 PAC pathway, RAS-ERK, PI3 kinase AKT, and other pathways not depicted here. Inhibitors that target kinases in these pathways are approved by the FDA or are in clinical development for various types of cancer. The Fernandez Valley Lab published a proof-of-principle ultra-high throughput screen of 1,280 pharmacologically active compounds that identified PI3 kinase as the lead target for therapeutics. This is not surprising, as the PI3 kinase and mTOR pathways are regulated by Merlin through association with PI3 kinase enhancer L, or PEG-L, shown by Wrong et al. 2004. Our lab has established a preclinical platform to screen and evaluate compounds for therapeutic development. Hits are compounds that promote loss of cell viability and induce cell death, screened in the high content and high throughput formats. 2 isogenic Merlin-deficient wild-type Schwann cell lines and 1 mouse Merlin-deficient Schwann cell line are used to evaluate the compounds in 2 viability assays and 1 cell death assay. The top performers are tested in the schwannoma allograft model. Phenotypic studies are performed, including flow cytometry and live imaging assays for cell cycle progression and cell death, Western blots for confirmation, and omics studies. Advanced compounds are studied in human primary vestibular schwannoma cells and confirmed in a second rodent model. Either the nude rat vestibular schwannoma xenograft model developed together with Christine Din at the University of Miami, or a GEM model for NF2 schwannomas. Our lab has created several pairs of human isogenic Merlin-deficient wild-type Schwann cell lines used in the studies I will present. Primary field Schwann cells are purchased from ScienceCell and are made Merlin-deficient by NF2 shRNA knockdown or CRISPR knockout. We also use a mouse Merlin-deficient Schwann cell line created by us from Schwann cells isolated from nerves from the NF2 exon 2 floxed mouse transduced in vitro with AdCre virus at passage 2. All 5 lines were used in the screen, and the HSO1 and MSO1 lines were used for phenotypic studies. The human cell lines are tested for loss of Merlin expression at increasing passages and express human Schwann cell markers HNA, S100, O4, and PLP. A 174 PI3 kinase pathway inhibitor library was purchased from Cellocan and was screened with 2 viability assays in 2 human schwannoma cells. 25 compounds were advanced because they had IG50s less than 5 micromolar and maximum activity greater than 50%. 10 of those were advanced because they were at least 2-fold selective for Merlin deficient over isogenic wild-type Schwann cells. Because we are interested in compounds that could promote regression of schwannomas, we now screen with cell death assays. Only 4 of the 10 compounds promoted a time and dose-dependent induction of caspase 3,7 cleavage in the HSO1 cell line. and only one compound had a nanomolar Ig50 and is in clinical trials. This is CUDC-907, and it was advanced for additional studies. CUDC-907 is a dual PI3 kinase HDAC inhibitor, also known as bimapinnostat. It is in 2 clinical trials for CNS tumors in children and young adults, which began in 2016 and 2019. It was previously studied by the Synnodos NF2 consortium and had efficacy in both schwannoma and meningioma cell lines. Results of the published schwannoma studies are shown. It has nanomolar Ig50 in 1 human myelin-deficient Schwann cell and 3 mouse myelin-deficient Schwann cells tested. It, however, did not slow growth of schwannomas in the periostin-Cree NF2 exon 2 flox gem model, nor did it preserve hearing in this model. I will present unpublished results from our lab and Dr. Din's at the University of Miami that demonstrate CUDC-907 reduces viability of 2 human myelin-deficient Schwann cell lines, reduces viability of 6 primary vestibular schwannoma cells in a dose-dependent manner, promotes apoptosis of human schwannoma cell models and primary vestibular schwannoma cells, and lastly slows growth of MSO1 cells by 44% compared to vehicle within 2 weeks in the mouse nerve allograft model. The library screen revealed that CUDC-907 has an Ig50 of 1 to 5 nanomolar, the lowest of the advanced compounds in both HSO1 and HSO5 cells. CUDC-907 has a high maximum effect, reducing cell viability by 83 to 94% in 2 assays. In MSO1 cells, it has an Ig50 of 0.66 micromolar and a maximum effect of 80%. Lastly, it has 5 to 111-fold selectivity window for human myelin-deficient Schwann cells over their isogenic wild-type Schwann cells. For Western blot analysis of target modulation, HSO1 cells were treated with 0.1 to 100 nanomolar CUDC-907 for 4 hours. We found that 10 nanomolar reduced levels of phosphorylated AKT at 39308 and serine 473, indicative of PI3 kinase inhibition. 1 nanomolar increased levels of acetyllysine, indicative of HDAC inhibition. Specificity of CUDC-907 is shown by no reduction in the levels of phosphorylated FAC or ERK. We conducted flow cytometry analysis of EDU incorporation to measure CUDC-907 effects on cell cycle progression. HSO1 cells were treated with 1 nanomolar to 1 micromolar CUDC-907 for 24 hours. 10 nanomolar CUDC-907 reduced the population of cells in S phase by 30% compared to vehicle, and increased cell populations in G1 and G2M by 20% and 10% respectively. To ascertain whether CUDC-907 induced HSO1 cell death, we conducted live imaging studies for 72 hours using a fluorescent cleaved caspase 37 assay. 2 staurosporin concentrations were used as positive controls, and untreated and DMSO-treated cells were used as negative controls. CUDC-907 was tested at a range of 100 picomolar to 10 nanomolar. Nuclear labeling with cleaved caspase 37 was detected in cells beginning at 16 hours of treatment and peaking at 24 hours. A dose-dependent induction of caspase 37 cleavage is consistent with induction of apoptosis. Cell death was confirmed in MSO1 cells using the same assay. Cleaved caspase 7 was detected in HSO1 cells treated with 100 nanomolar CUDC-907 for 30 hours by Western blot. Cleaved caspase 3 was not detected with the antibodies used. To confirm these results, we conducted violet ratiometric assay for membrane asymmetry analyzed by flow cytometry of HSO1 cells treated for 24 hours with 1 nanomolar to 1 micromolar CUDC-907. 100 nanomolar CUDC-907 promoted the appearance of apoptotic HSO1 cells by 24 hours. Our collaborator at the University of Miami, Dr. Christine Din, a neurologist, consents patients undergoing resections of vestibular schwannomas for use in translational studies. Cells from 6 vestibular schwannomas were expanded in culture for 1 to 2 passages and then treated with 4 concentrations of CUDC-907 and 50 micromolar rapamycin as a positive control for 72 hours. Viability was measured using cell titer glow. All 6 vestibular schwannoma cells studied thus far have had a dose-dependent decrease in viability beginning at 1 nanomolar. 100 nanomolar CUDC-907, the highest tested concentration, reduced vestibular schwannoma cell viability by 50% compared to DMSO-treated cells. CUDC-907 is the first compound identified during the last 4 years that reduces viability of all vestibular schwannomas studied. 3 of the 6 vestibular schwannomas were treated with 0.1 to 100 nanomolar CUDC-907 for 36 and 48 hours, and assessed for induction of cell death with a caspase glow 37 assay. 50 micromolar rapamycin was used as a positive control. All 3 vestibular schwannoma cells showed cleavage of caspase 37 at 100 nanomolar at 48 hours. CUDC-907 is the first compound studied during the last 4 years that induces apoptosis in all these vestibular schwannomas studied. The in vivo efficacy of CUDC-907 was assessed using a sciatic nerve allograft model of NF2 schwannomas. This schwannoma tumor model has been used for preclinical testing of bevacizumab, rapamycin, and crizotinib that are in clinical use for NF2. Our lab has used this model for other preclinical studies. 5,000 MSO1 cells expressing luciferase were implanted into the sciatic nerve of 17 immune-deficient NSG mice and allowed to grow for 7 days. 9 mice were treated with a vehicle and 8 were treated with CDC907 at 25 milligrams per kilogram for 14 consecutive days by oral gavage. Mice were imaged using IVUS at 7 and 14 days of treatment. 25 milligram per kilogram CDC907 slowed implant growth by 44% compared to vehicle at 14 days of treatment, as measured by radiance. Immunohistochemical analysis of the graph revealed a statistically significant decrease in phosphorylated AKT and higher acetyl levels in treated mice compared to vehicle controls. This demonstrates inhibition of both PI3 kinase and HDAC in vivo. There was, however, no significant decreases in Ki67 or cleaved caspase 3 levels. Testing of cleaved caspase 7 is in progress. In conclusion, CDC907 is the top-performing inhibitor from the 174 PI3 kinase pathway library screen that met viability and cell death assay criteria in human schwannoma cell models. CDC907 slows implant growth by 44% compared to vehicle in the schwannoma allograft model. Moreover, CDC907 induces a dose-dependent loss of viability in the 6 vestibular schwannoma cells examined and promotes cell death in the 3 vestibular schwannomas studied. CDC907 shows prevalent HDAC activity matching kinome data from the Synodose NF2 consortium. Our results suggest that fimipinostat has the potential to promote schwannoma regression in NF2 patients. We are working to obtain fimipinostat from Curis Inc. We intend to repeat many of the studies presented here with fimipinostat. Lastly, we will test efficacy of fimipinostat in a nude rat via xenograft model published by Din et al. 2018 that assesses hearing and balance as well as implant growth. Thank you for listening to my talk today. I would like to thank Dr. Vaya and her lab, Dr. Hugel, Dr. Petrilli, Rosa, Parth, and Michael for their contributions. Thank you to the DIN lab at the University of Miami for their collaboration on this project. These studies are funded by the NIH.
Are you including apoptosis as a screen? Benign tumors may not need that.
So in our preclinical platform screen, as I mentioned earlier, we've included cell death as one of our screens because we're interested in tumor regression and not just cell cycle arrest or stopping of growth. So we've utilized a cleaved caspase 3/7 assay to do that, which I think can be indicative of both apoptosis and some forms of cell death.
Yeah, wonderful. And here is another question.
Matthias, can I comment a little bit?
Oh, yes, please, Christina, go ahead.
I'm sorry to butt in, Maria. I think it's important to look at drugs that will cause apoptosis of these benign schwannoma cells because clinical experience has shown that as soon as you take pressure off the tumors, cells regrow. So there's yet to be, to my knowledge, a therapeutic that will promote regression of a benign schwannoma and for the long time. So we're hoping that we could kill off some of these benign schwannoma cells that are already growing and impacting function in the patients.
Excellent. And I have another question here from Liam Laraba. And the question is, did you confirm that the primary vestibular schwannoma cells were NF2 null?
Yes, so genotype testing is in progress right now, so we don't have those results quite yet.
Okay. And let's see if there's any more questions. No, but I would like to ask a question. We have some more time, so if that's okay. And the question is, well, there's 2 questions. One actually would be regarding, are you planning to do any additional mechanistic studies to, dissect the mechanism of action further? And then number 2, as you may know, combination therapy is, often difficult to realize the clinic due to toxicity, but of course we're always looking for, additive therapies. If a single agent may not be sufficiently effective. So one combination here that one could think of that could have non-overlapping toxicity could be radiation. So I was wondering if you also have any plans or perhaps the idea of of testing this agent in terms of synergy with radiation, perhaps like low-dose radiation, and how these might work together?
Yes, okay, thank you for the questions. In terms of your first question in relation to mechanism, yes, we will be moving towards those studies next. We are not currently looking at anything specific, but we have some targets that we could be investigating next, especially following obtaining vitipinostat. In terms of radiation, that is a great question. I'm not sure I have a great answer for that. Our collaborator at the University of Miami, Dr. Christine Din, often does studies with radiation, so she would be someone to consult with about that. So it's a good thing to consider.
Okay, wonderful. There are no additional questions coming in, so we're going to switch to the next and final presentation of this session. And this is going to be by Haley Hardin, also from the Fernandez Valley Lab. And she is going to talk to us about preclinical assessment of combinatorial PI3 kinase mTOR and FAK-SRC pathway inhibitors for treatment of NF2 schwannomas.
Good afternoon, and thank you for the opportunity to present our work on evaluation of a drug combination for NF2 schwannomas. I'm a first-year grad student in the Fernandez Valley Lab at the University of Central Florida. We have no conflicts to disclose and will discuss an FDA-approved drug. I will briefly review work by the Synodus NF2 consortium that served as the foundation for this study by identifying SRC and FAK as kinases to co-target with PI3 kinase mTOR inhibitors. Since then, our lab has identified omepallisib and dasatinib as a combination for further evaluation in human HSO1 cells, a mouse sciatic nerve allograft model, and in vestibular schwannomas. In 2018, Synodus NF2 published results on omepallisib, a dual PI3 kinase mTOR inhibitor that reduced viability of human and mouse Merlin-deficient cell models. However, it was not effective in vivo in the Perry-Austin-Cree NF2 mouse model at the doses tested. It was postulated that this was due to activation of compensatory pathways. Kinome studies by Gary Johnson's group found that HSO1 cells treated with omepallisib increased activity of FAK, shown here by its gene name PTK2. This did not occur to the same degree or at all in isogenic wild-type HS11 cells treated with omepallisib. Similar kinome results were found in an isogenic pair of mouse Merlin-deficient and wild-type Schwann cells created by Helen Morrison. The Merlin-deficient cells differentially increased activity of FAK in the SRC family kinases SRC and LIN in response to omepallisib. These results supported co-targeting PI3 kinase mTOR and FAK-SRC pathways for schwannomas. This is a more comprehensive but still incomplete summary of known Merlin-dependent pathways. In Schwann cells specifically, FAK is activated by integrin receptors adhering to laminin in the extracellular matrix. FAK plays important roles in Schwann cell proliferation, cell cycle exit, and myelin gene expression, pathways this lab has contributed to mapping since the 1980s. In Schwann cells, ERB2 and 3 receptors bind neuregulin, a major Schwann cell mitogen and regulator of Schwann cell motility during active myelination. ERB3 activates PI3 kinase through a direct binding site. Others have shown that FAK and YAP activity increase in Schwann cells with loss of Merlin function. The Synodus findings suggested that omepallisib increased SRC-FAK signaling to promote cell survival. In cancer cells, FAK activity has emerged as a major contributor to drug resistance. It is activated in RAS and RAF mutant cancer cells treated with RAF or MEK inhibitors and promotes cancer cell survival by activating Wnt beta-catenin-dependent survival. 2 FAK inhibitors are in clinical trials for cancer in combination with MEK and RAF inhibitors. Lastly, FAK promotes nuclear translocalization of YAP. YAP activation correlates with grade, metastasis, and poor outcome in many cancers and is implicated in the development of drug resistance. We conducted a combination matrix screen of a curated library of PI3 kinase and FAK-SRC pathway inhibitors approved by the FDA or in clinical trials. 4 human and 1 mouse schwannoma cell models were used. Results in the HSO1 and HSO2 cells are shown here. The lines differ only in the short hairpin RNA used to decrease Merlin levels. The inhibitors were tested at 10 doses. Drug synergy was evaluated using 3 mathematical models. The Lowy synergy is shown. Omepallisib and dasatinib had the highest synergy sum score of 83 and 56 respectively. In HSO1 cells, the highest synergy occurred with omepallisid at doses between 3 and 16 nanomolar, well below its IG50 of 50 to 150 nanomolar, and with dasatinib at doses between 100 to 600 nanomolar, well below its IG50 of greater than 10 micromolar in HSO1 cells. Dasatinib is FDA approved for treatment of specific types of leukemia and is in combination trials with other drugs. Omepallisid has been tested alone for pulmonary fibrosis and in combination with the MEK inhibitor trametinib for solid tumors. We conducted a live imaging study of HSO1 cells grown in the presence of omepallisid and dasatinib alone and together at 3 concentrations. Proliferation was assessed by well confluence, and cell death was measured with a fluorescent cleave caspase indicator. Only well confluence results are shown because no condition induced caspase activation. There was a synergistic and dose-dependent decrease in cell proliferation for the drug combinations compared to single drugs at the same concentration beginning after 8 hours of treatment. At 72 hours, cells treated with the highest dose of each drug showed that OMI-treated cells were 52% confluent, DASA-treated cells were 60% confluent, and cells treated with the combination were only 17% confluent. Target modulation was assessed by Western blots. Both drugs reduced levels of expected targets. Omepallisid alone, at both doses or in combination with DASA, reduced levels of phospho-AKT, threonine 308, and serine 473. It also increased phosphosarc levels at these low concentrations. Dasatinib alone at both doses reduced phospho-FAC and phosphosarc levels. Dasatinib alone also decreased levels of phospho-AKT at threonine 308, but not serine 473. The in vivo efficacy of omepallisid and dasatinib each alone were assessed as part of larger studies. 5,000 MSO1 cells expressing luciferase were implanted into the nerves of NSG mice and grew for 7 days, followed by 14 days of consecutive oral gavage. After 14 days, omepallisid-treated mice had a 33% reduction in fold change increase in average radiance compared to grafts in vehicle-treated mice. After 14 days, dasatinib-treated mice had a 32% reduction in fold change increase in average radiance compared to the grafts of vehicle-treated mice. To determine the efficacy of the combination, a small pilot study was conducted with 9 NSG mice. The mice received 20 milligrams per kilogram of dasatinib and 2 milligrams per kilogram of omepallisid for 1 week, then 1 milligram per kilogram for the second week due to toxicity. After 14 days, grafts and combination-treated mice had a 42% reduction in both tumor weight and fold change, increase in average radiance compared to vehicle. These results indicate that there is no antagonistic effect of the combination. However, a larger study is needed to determine if the combination is superior to either drug alone. Our collaborator, Dr. Din, cultured cells from 7 vestibular schwannomas. Some VS had areas of different textures and were cultured and tested separately. All VS had NF2 gene mutations in the tissue, only one was from an NF2 patient. After 1 to 2 passages, cells were seeded in 4 replicate wells and treated with 3 concentration of drugs alone and together for 72 hours. Viability was measured using cell-tethered glow. A statistical analysis of the data, conducted by grouping all the vestibular schwannomas per condition, revealed a statistically significant reduction in viability compared to DMSO controls. However, the combination was not superior to single drugs. The variability in response to the drug combination is shown here, with each vestibular schwannoma plotted separately. 3 of the 7 VS cells had a partial response to omepallisid and dasatinib combination at the lowest dose. Some of the VS decreased viability at higher drug combinations, but there was not a consistent response amongst the vestibular schwannoma cells. An example of a responding vestibular schwannoma is VS21. It had a loss of viability to the low dose of omepallisid, below the IG50 in our human model schwannoma cell lines, as well as a surprising response to dasatinib in a dose-dependent manner. The 2 lower doses of the combination were statistically superior to each drug alone at the same dose. The viability response could be predicted from the expression levels in the native tumor tissue of both phospho-AKT, serine 473, and threonine 308, as well as high levels of phospho-FAC and phosphosarc. Cells from the NF2 vestibular schwannoma surprisingly responded to omepallisid alone at the lowest concentration. Co-treatment with dasatinib blocked the response to omepallisid. At the highest dasatinib concentration, there was some reduction in viability that likely contributed to the activity of the combination. Surprisingly, we did not detect significant levels of phospho-AKT nor phospho-FAC or phospho-SARC in this tissue. Thus, better biomarkers of drug response are needed. In conclusion, sub-IG50 concentrations of each drug in combination reduced viability of 4 human and 1 mouse schwannoma cell models. Apoptosis was not induced. Each drug alone and in combination in separate studies slowed growth of MSO1 cells and mouse nerve allograft models by 33% of vehicle controls. In 7 vestibular schwannomas studied, there was heterogeneity of tissue texture and responses within and between the VS. Low doses of dasatinib or omeprazole alone reduced viability of some VS cells. Levels of phospho-target expression are not always predictive of drug response. The drug combination was superior to single drugs in only 3 of 7 VS studied. We are working with Dr. Din to optimize a precision medicine approach for vestibular schwannomas and are reconsidering the value of a larger in vivo efficacy study with this drug combination. Thank you for your attention. I'd like to thank Dr. Valle and her lab, Dr. Hugel, Dr. Petrilli, Rosa, Abdul, Michael, and Parth for their contributions. Thank you to the Din Lab at the University of Miami for their collaboration on this project, Dr. Kara Hannes for discussions and guidance on drug selections. These studies were funded by the NIH and DoD. Dr. Fernandez-Valle will be answering any questions.
If there's any questions coming in, while we wait for this, I'm going to ask a couple of questions. One concern, of course, as we treat these patients is secondary resistance, right? We might treat them with a drug that is modestly active, but then, eventually the tumors will continue to grow. And there may be obviously many mechanisms that govern that. And so far, I think that has been, not really looked at very much, at least in the published literature and what I've seen presented. And so my question is, because combination therapies are often difficult to realize in the clinic because of toxicity, is there potentially an avenue for studying resistance mechanisms to secondary resistance mechanisms to modestly active drugs that, some of which we already know and additional ones that you have identified, see how secondary resistance developed and then potentially, address that, either not necessarily with a combination, but perhaps also with sequential therapy where you might, instead of combining 2 drugs at the same time, use them sequentially, but do it in a smart way based on, resistance mechanism and treatment evasion, sort of strategies. So I was curious, what do you think about that?
Yeah, that is a great question. And looking into acquired resistance to certain treatments that are already approved, either for NF1 or in clinical trials for NF2, is something that we are actually going to start working on in our lab, something that we're looking forward to and thinking about. I think it's a great question on whether we should pursue combinatorial drugs or sequential treatment. And I think that's definitely something that should be looked at more in these kind of drug studies, both treatments together and one right after the other, and which could be more effective and translated into clinical research. So it's definitely something that we're going to be keeping in mind and working on.
So Matthias,
Yes.
if I can say something about the use of FAC inhibitors. So there's a review that just came out recently, I think a few months ago, on the use of FAC inhibitors in many types of cancers, because it is a primary cause for drug resistance. And what they're finding is they're hoping that they can just treat patients with lower doses of the primary drugs so that you could perhaps even decrease development of resistance. You and I have had conversations in the past about whether sub-therapeutic doses make sense to treat a patient. Whereas I'm reading that now, it might be a possibility, especially some of the compounds that famipinostat that have some type of toxicity. HDAC inhibitors have toxicity, but I know some of these are now in clinical trials, at least early phase 1, and seem to have tolerable side effects.
Terrific. All right, thank you. And there's one more question coming in. We have time. So this is again from Oliver. How do you select drugs to take forward to clinical trials?
So that is a really good question, and I'm not 100% sure. We mostly work on the preclinical side, but we do work in conjunction with physicians and see what they're interested in, see what exciting results we have, and we are in discussion as far as exactly how those get chosen. I think Dr. Valle might be able to speak a little bit more on that, or maybe even you, Dr. Karahanis, than I would.
Christina, you want to say something, and I'll be happy to say something as well.
All I know is that we have this pretty robust platform where we can, start from a drug library that we can repurpose compounds or novel scaffolds. So if you're starting with the repurposing FDA-approved compounds, all we can do is say, this is what's working on our patient vestibular schwannomas. We're very interested in the CUDC 907 because that's the only drug that Christine Din has found that in every vestibular schwannoma she studied had a consistent response that was dose dependent. We've only looked at 7 vestibular schwannomas, but we can increase that sample size. And we know that if it works in our mouse models, we then pass the information on to her, and as she resects the tumors from her patients, most of the patients are sporadic vestibular schwannomas. She tells us what works, and then we talk to Matthias and Scott and other clinicians, and it's really up to them.
I'd be happy to expand on that. In that, which drug do you take to a clinical trial? It really depends on a variety of factors. The first factor, of course, is whether the drug is even available, right? Sometimes there are drugs that are being developed by pharma, or sometimes even entire classes of drugs that then really don't make it to the clinic. And they may not make it to the clinic, not because they don't, quote, work in NF2, but they don't make it to the clinic because they fail in cancer clinical trials. And as a result, the company will shelve the entire compound, or the entire class of compounds will be, shelved, so to speak. There's no more clinical development. And, unless someone buys the patent and starts a new company, these drugs are, so to speak, dead. And, even if you have good data for NF2, it would be very difficult to develop those, and bring those into a clinical trial. And then, drugs that are either approved, that are approved for an existing indication. Of course, those are relatively, easier to take to the clinic, depending on whether there's, whether they're, still patented or whether they're generics available, will influence potentially that decision, and whether the company, would support a clinical trial, because if the drug is not FDA approved, then you would have to do this under an IND application, and then it takes additional regulatory steps. So really, long story short, it really depends on the clinical development status of that particular drug or combination that you're interested in. Whether these are drugs that are still active, whether these are drugs that already have human data or not. Otherwise, you would have to start with a phase 1 toxicity study. So ideally, you would want to do that with drugs that are more advanced in clinical development, and all these considerations, will kind of govern of, what you can actually bring to the clinic. And that funnel, of course, gets narrower as you get closer to the clinic from what you can, screen in a, large, high throughput preclinical screening platform. All right. Well, thanks to everyone who contributed. This was a terrific session, and we are ready a few minutes before the official end. So this is great. We'll give everyone a few minutes back to enjoy the next sessions. Okay. Well, thanks everyone. It's been a pleasure. Thanks to my co-chair, Miriam, and thank you all for contributing to this terrific session.